Great. It's my pleasure to open up the last session of the day before the poster session. Especially, it's my privilege to be introducing four longtime colleagues and friends. The first of which is Jian Kang, who is a student I co-supervised, and now we are collaborators. And it's a pleasure to continue working with him on large-scale Bayesian models, and that's part of what we talk about here.
All right. So you can hear me, right? So yeah, thanks, Tom, for introducing me. And it's my great pleasure to co-organizing this conference with Hongtu. And thanks, Hongtu, show me this opportunity like 1.5 years ago. And this is wonderful that IMS and SF can support us and to come together to discuss this research topics. So yeah, this session, our topic is the population neuroscience, and I will basically exactly follow the format that Hongtu suggested, that just discuss several questions and using some of my experience. So this is challenges and opportunities about the population scale neuro imaging data analysis. So basically, what do I mean by population scale neuroimaging data analysis? So basically, the population here, I emphasize on large groups of people, but of course, in statistics, the population is a relative concept versus sample, right? So it's your targeted population. And here we are thinking about more like we have a very large scale population, a large group of people. And the basic question here is try to understand the brain structure and function vary among individuals within the population of your target population. And how these variations are related to various cognitive or behavior or clinical factors. So I borrowed one figure from Hongtu's review paper. This is a very nice review. I suggest each of you to read this paper. But this figure basically summarized some of the large studies focus on different population, varying different the age range are varying, and many of you already illustrated your method or your data analysis using those studies, and I will talk about ABCD and UK Belbam as well in my talk. So I start from this question that what would be the most important problems in the future, in the near future, basically. I think I don't have a lot of new things here. Many of you already talk about like reprocessing large scale data. So here I emphasize on we need to think about how to preprocess large scale data. It's not that who gonna do it. It's that when you know how we should do it, right? Well, what would be the right way to do it, and when you have clear about your goal of your analysis, so you probably need to think about the correct procedure or like the better procedure to preprocess data. And another important problem is how to increase the scalability of the computational intensity method. I think the success of the ChatGPT because it has that scalability of the pre-trained transformers and can handle that very large scale data. And I think this itself is actually the statistical problem. So how you actually train your model to handle very large data, extremely large data set. And you can be either frequency or Bayesian, no matter. And Bayesian may be more computational intensive, but actually deep learning methods also require a lot of computing resource. So and also in the population scale neuroimaging data analysis, we need to address the heterogeneity, which is very become more and more common in large scale imaging studies. That's a different type of heterogeneity. I will illustrate some of our experience with that. And for traditional analysis for the targeted distribution, maybe we can think about how to reduce sampling bias, how to do the weighting for the large scale population analysis. And another topic is about integrated multimodal imaging data. But I only have 15 minutes, so I will just talk about some of our experience and illustrate here a little bit. So here is the work joined with my former PhD student. She is now in Duke University as a postdoc, and also my former PhD advisor, Tim Johnson, and mentor, Tom Nichols. So this is basically we are trying to apply fully Bayesian analysis on the very large scale data set. Specifically, it's UK Biobank task fMRI imaging analysis. And the key point here is that we want to integrate the subject-specific mask, which is quite unique for large population studies. And also, can we develop scalable posterior computation to do better inference and compare with like a simple method like a massive univariate analysis? You just ignore those like missing values and how the performance can be improved. So yeah, this is the basic motivating data set that we look at. Particularly the question of interest is the influence of age on brain activity across the whole brain. And we consider this Image-on-Scalar regression framework, and the response variable is the emotion fMRI task contrast map, and primary covariance of interest age. We definitely incorporate a few other covariance. We did not actually run into genetics data yet, but the idea here is that we just look at the age, but adjust for other factors. And this is a large data set that we look at almost 40,000 subjects, and we have the fMRI contrast map actually after pre-processing steps, and we get this like 2 millimeter register everyone into 2 millimeter MNI space, and we get a very large data set is over like 180 gigabyte. And so one issue about the pre-processing that if you carefully pre-processing each subject individually from the both time series signal to this contrast map, you will have individual specific mask, because some voxels for each individuals may not actually reliable. You can remove that during the pre-processing steps. Then you actually come up individual specific mask that you trust that voxel value is well reflected the contrast map, contrast statistics during the fMRI analysis. So then when we look at this intersection mask of over 40,000 subjects, I think Tom also showed this morning that is about like only 28% of the voxels actually have a common locations. So basically, if you think about the simple way to do that, you consider this way as one of the step of the pre-processing, you probably will just either remove all other voxels or like just impute them with zero. So that's a lot of different pre-processing steps you can consider as pre-processing step. But if you think about this as part of your statistical problem, that is not only pre-processing step, like we need to think about how we actually deal with this issue. And if you run some simple method, you may lose power to detect signal. So here is the figure showing that that's actually the purple area is 100% of the blue area actually is about at least 50% observed. So we go with this Bayesian scalar image-on-scalar regression. So we have a subject-specific mask. We have activation voxel selection indicator. We have a spatially variable coefficient, and also take into account subject level spatially varying effect. And we assign a Gaussian person parameter. So the model itself seems like complicated, and but if you have a small data set, the full Bayesian computation is actually quite straightforward. If you took like a basic applied Bayesian inference, if you have small data set, this type of model is quickly can be run by, if you use Stan, use JAX or IMC, that can be done easily. But if you have that large scale data, you have UK Biobank level data. So this is not easy. So basically you have a 180 gigabyte data. So you cannot run fully Bayesian inference easily. So this is what we try to do. But the point, another point here is that we have an individual specific mask here. So that is actually quite hard to handle if you consider some other type of like potentially imputation methods separately. That also very computation intensive. So here we have some strategy to do that. So we have this scalable posterior computation algorithm proposed. So first we do some like a basis expansion approach to represent the model, do some like transformation. I won't skip all the details, but if you want, you can look at our source code and our paper. The idea here is that we need to take advantage of this Gaussian perfect prior. Basically, we assume the spatial information will help us to borrow strengths in neighboring voxels, and then we can reduce the dimension. And we need to learn some, we need to use some like relatively advanced computer programming technique that include memory mapping, and also we are trying to using this MCMC algorithm, which is based on the stochastic gradient long dreaming algorithm, which is scalable, only take the mini batch of the data over iterations. You don't have to take all the data over iterations. This is actually a huge improvement on the scalability of your algorithm, which has been also widely used in like a deep neural network. So which is the technique that you can actually learn from the deep neural networks that or deep learning field that can be analyzed for your data set as well. So here are some like evidence that this method actually can give you better performance. And so basically, if you control FDR as that, if you compare the massive univariate analysis, if you just do the regular Gibbs sampler, you can see the power has actually improved a lot. If you simply just impute zero values, then actually you lose power. And you also here as you can see, we compare the computation time and our scalable method, which is much faster than the regular Bayesian method. I did not list the MUA is still the lightning fast, as Tom mentioned. The computing time we cannot beat that, but you can see the memory cost. We actually, if you use scalable method, if you carefully coding your software, you actually can have much less computing power, much less memory cost actually compared with the massive univariate analysis. And here are some results. We can see that we actually for this UK Bellband data, we apply our method. We do some pre-processing after on this contrast map, and do much better job on the memory control the memory use. We only need a 14 GB memory to run our program, and about like 20 CPU hours. And if you do parallel battery, you can speed up the computing for this type of inference. And what we gain here is that you can compare the massive univariate analysis, and we can identify much more like the activation region compared with massive univariate analysis. And there's another example I probably don't have time to show. It's more like how you address the heterogeneity of our population. So here we want to emphasize there's a population heterogeneity associated between the brain activity and the individual characteristics. So we actually performed some data analysis on ABCD study, and this work primarily done by my PhD student who just graduated last week, Zikai Lin, and my long-term collaboration, Jian. And the whole idea here is that we introduce like a subject-specific regression coefficient, and then we assign that as a group. Potentially, those subject-specific regression coefficient may take a finite number of distinct values. So basically, you can actually do clustering of this of the regression coefficient. It's a sort of extension of the finite mixture model in the regression setting, but our outcome of the regression is an image. So, and I will skip the details, but basically, when we carry out the analysis on the ABCD study, we find out that we can find 4 different distinct groups, and then we compare the analysis. You only assume there's only one group, so we will see much improvement in terms of like within group, like projected MSE, the mean square error. So basically, we fit data much better, and the 4 groups actually have a quite different kind of characteristics. So that's another direction that maybe you can think about a better method to handle this type of heterogeneity, and you can consider in the future. So, and here is another question that I want to try to answer is how you pre-process data potentially with AI. And so ideal path, you can just think about it, that there's AI, you can do the raw data directly to pre-process data. This is not possible. So I think what you can do is more like how to using the raw data and actually using AI to summarize features of the raw data and that is related to the operation and the pipeline, and also using AI to generate code or using AI to generate and then pre-processing that. So there's a lot of steps you need to decompose that and train AI. So that I think that's has some potential you can use AI to do that. And for the data sharing, there's a platform. And I think we need a platform, but I think many ideas has been discussed. I will not reproduce here. I think one thing I want to say is that you want to introduce your method to your collaborators, and potentially you can do social media, and also try to organize conference and the workshops and invite people in imaging computing to attend. And also, I think many successful statisticians, they join and lead a research team to design, study, and collect data. Otherwise, I think that's also important. Yeah. Thank you all.
Thank you. I will save questions to the end. So our next speaker is Liza. Also, so Liza's the chair of the statistics department. He's been a long time contributor to methods for network data.
All right. Thank you for the invitation and the introduction. My pleasure to be here. I'm not really a neuroimaging person. My primary expertise is in network data analysis and covariance matrices. So I thought the only thing I could think to do with 15 minutes is sort of walk you through how my thinking from that perspective on neuroimaging has evolved and give some examples from prior work of the path that we took in this area. I'd like to thank my collaborators. My two primary science collaborators are both in psychiatry. They're both MDs, so we focus on humans. Former PhD students and postdocs in statistics, and a remarkable person who was first in psychiatry as research staff, and then my PhD student, Dan Kessler, who I think more of you in this room probably know Dan than you know me. He's who made it happen because he could go in between us and the funders. So in bird's eye view, mainly, not all of my work, but mainly my work has been focused on fMRI imaging, mainly resting state, and applications, either mental health disorders or developmental applications. They're also interested in kids. I think of them as weighted networks. And when I say networks, I mean the whole brain. I don't mean subsets. There was early work where people tried to threshold correlations obtained from fMRI imaging and turn them into binary networks, which is zeros and ones. That really didn't work. I think that just lost way too much information. So we don't do that. Now I mostly work with the standard pre-processing pipeline that my collaborators use on a standard parcellation, standard atlas they map it to. I'll say a little bit more about how we ended up there. The scale is usually like a few hundred ROIs in these atlases. My main focus is on the relationship between the neuroimaging data, so the brain connectivity matrices or networks, whatever you want to call them, and subject level covariates. You can have one covariate, you can have multiple covariates. They could be categorical, like disease status, or they could be continuous. And we've looked at it both ways. We've looked at predicting images from covariates, predicting covariates from images, and generally just trying to understand the relationship. So that the errors could go either way in this. Challenges, mine are not different from all of yours. We all know the same kind of trouble we have, this, especially with fMRI data, very noisy. Samples are getting bigger, but we started out very small. They're still at best moderate. And preprocessing, my focus is on producing interpretable results that somebody can tell a story about. I think prediction by itself is not that interesting in this context. And what I mean by network-aware analysis, and I know that my use of the word network is maybe not standard, is think of inference at the right scale. Nobody's really interested in an ROI, as far as I can tell. Nobody's really interested in one edge connecting one place to another. What people are interested in is something like the default mode network, prefrontal cortex, or communications between these things. I'll call them systems to distinguish them from networks. So what I call network cells here is any block in this connectivity matrix. So it could be a communication within one system or communication between systems. All of these things are cells. And that, I think, is the scientifically interesting scale, but it's also a tricky one because, I mean, as you can see, even in this picture, it's quite heterogeneous within a cell, and you don't want to just go an average because you get a lot of signal lost in that. And there we have various trade-offs in scale. So to start with individual edge weights, they're not very interpretable, but that's sort of the raw data. That's where, well, not raw, but pre-processed, but that's where the signal is. So in a way, you have the most information if you stick to edges. People commonly do PCA, ICA, some sort of projection, denoising of the matrix. I initially didn't want to do that because that loses interpretation. I've come to see that the gains on reducing the noise are also very substantial, so it is worth doing. So this helps with noise, but loses interpretation. Then the global network summaries, that's popularized by Olaf Spurns and people like that. They were very popular to start with, very interpretable potentially, but I think often too crude. There are just not enough of them. There's maybe like 5 numbers that will give you a global summary, and that's just not really enough to get an interesting answer. So this is kind of an increasingly global in resolution. And so cells are supposed to, if you can, get somewhere in between. Be interpretable, but still try to reduce noise and dimension. So here's a first example of first attempt to do something like that was modeling the connectivity matrix. So this is the connectivity matrix. You don't need to pay attention to this equation. It's just for illustration. Some kind of mixed effect model. There was a cell level effect and there was an edge level effect. So you could decompose. And then you focus on interpreting that, and the edge effects they help you predict. That was the idea. We didn't want to throw them away entirely because they still help you predict. But you don't care about their coefficients because that's not the right scale. And the cell structure was incorporated in both mean and covariance. So there's also dependence between cells that was allowed. So that was reasonably nice, but still like modeling the weight of one edge. That wasn't quite right. We did it also the other way, looking at predicting a covariate from connectivity. In the case of this paper, disease status. So it was a network classification paper. That okay, we'll look at all these matrices. We'll try to classify them as people who have this study was schizophrenia, people who have schizophrenia versus people who are healthy. Yes, we can do that. We could predict it. Accuracy is not too bad. But then what do we learn from that? Because at that point, we were still networks people more than neuroimaging people, and we were trying to keep it as a general networks classification, we tried to select active edges and nodes. So not just edges as variables, but also nodes. That helps, gives you a nicer picture. Still, ROI is not quite there. Then we thought, okay, well, we can do better than that. We can do, I think a lot of statisticians go through the thought process, start doing this. There's some standard pre-processing, standard atlas, and you think, surely we can do better than that if we do our own parcellation. And we can. The question is for what? And what are we losing by doing that? So this was another idea that we had. We said, instead of using standard parcellation, let us learn the best parcellation for this classification. We're still going to do it at the cell level, but we will learn the best brain systems. Maybe we'll discover something. If you look at this comparison, this is in terms of classification accuracy. If you just throw all the edge weights in, you actually get the best accuracy, because that's where all the signals. You're not losing anything. You're not constraining. Fixed systems from a standard atlas, not so good for this problem, 60% accuracy. And then, if you do some selection on edges, doesn't really help. But if you learn the systems, you're still at the systems level, but you get not all the way to that accuracy, but 80%. So you improve a lot. So, okay, great. You've learned some systems, but what do they look like? This is what they look like. This is a diagram comparing a standard atlas to the learned systems. Spatially, they're mostly contiguous. If you plot them on the brain, you think, okay, that's not completely random. It's probably not complete junk. But what do we say about them? Like we can say, for example, that this is the DMN. It got split into 3 systems. You have different signs. That's something. But in the end, you end up with a picture that looks like this, and you don't really know what the scientific story is from it. So even though we could do this and improve classification results, and we also have an extension to multiple responses where you can do that, in the end, we went back to standard atlases just so we can compare to other people. Another example, also cell level group penalties. Mostly we do this through penalties. Actually, let me skip this one. Talk a little bit about the current focus. People have already introduced the ABCD study, so I don't need to talk about that. Michigan is one of the sites, and so we work closely with the people there. My current focus is a lot on multitask learning in this context. Multitask learning is the machine learning term. In statistics, people call it multiple response prediction often. It's just a vector valued regression response. If you do this jointly, if you predict your correlated responses jointly, and you leverage the correlation between them, you should be able to do better than if you predict each one separately. Often this is a second order effect, not the first order effect. Often it's you improve the variance, but still, clearly there's information there. So focusing on that, sort of gave up on using the edges and started working with PCA components because there's the trade-off between denoising, interpretability. It's like all goes around this thing. And what I'd really like to do is inference. And that is very hard, right? But we as statisticians, this is something we could bring to the table that other people don't do. Longitudinal modeling is future work, and now that Tom gave his great talk, I can just go read his papers and done with that. But so here's one example with multiple responses. This is from ABCD study. This is a cognitive task battery. It's 9 different tasks that they're given to do. The kids in the study are given to do. So you have these 9 scores. So if you look at them all together, and some of these questions relate to general intelligence versus specific intelligences, and there are all sorts of other issues I'm not going to go into. But if you look at all of them together, what can you really say? One of the things we did is look at, this is another way in which we went away from prediction, is, okay, you could predict the brain from the covariates, you could predict covariates from the brain. Let's not commit one way or another, just look at canonical correlation. That's symmetric. And that just means you give it one set of vectors. So one set of vectors is the scores, another set of vectors, that's the matrices after PCE denoising, and you look at most correlated projections, basically. There's old school inference for that, like from the 50s, which is only for low dimensional, only for Gaussian, only of the same size vectors. So definitely the brain and the task battery are not the same size. So we did something computationally. We did bootstrap inference. And specifically for directions, correlations we can also do, but that's much easier. Directions is what you really want to know about. Technically, the main challenge in doing bootstrap inference here is alignment across bootstrap replications. I'm not going to talk about that. But what kind of picture can you get? Like, I don't want to go into the details, but this is the kind of thing that where I feel like this is something statistical that we can bring to the neuroimaging world that they don't necessarily do. So this is the first three canonical directions. So sort of like the most important things. And then the red ones are the ones where the bootstrap confidence interval doesn't cover 0. So it's according to bootstrap inference, these are significant. So you can look at which task shows up at which component. So you see they're distinguished. And you can also see that some of them really just pick up on one. So there's something being isolated. How you interpret it further is an open question, but at least you have a more or less rigorous measurement, more rigorous than variable importance, right? Because at least we can check whether it covers 0. You can do it in more model-based way. This is a paper that just came out in AOS. Like always, there's a trade-off. Selective inference it's been very popular in statistics over the last 10 years. That's what they call honest inference after model selection. We can do this here as well, but then we need a linear model and we need Gaussian assumptions and we need, more selective inference things. So again, a trade-off. You get, in a way, you get better results because once you made your assumptions, I'm not going to go into the details, but the main thing is your confidence intervals shrink. You really are leveraging your correlations, but you made the assumptions. And so you're doing better, but what does that really mean? So I won't separate into the 3 questions, but I'll just share some thoughts generally. I think reproducibility and stability are crucial. And for that reason, we went away from trying to improve pipelines to using standard ones, because that's easier for people to compare. I think we can improve our validation pipelines, which will help reproducibility. I think our friends in genomics are actually ahead of neuroimaging on how many sets they test on, how many standard data sets they have. So we could do that. Pre-processing, one small point I just want to make is that I say using the standard pipeline and they are getting better, but there's never going to be the right way to do pre-processing. There's not going to be one right answer because a lot of the nuisance variables that get taken out during pre-processing are correlated, are confounded with the response. In my setting, a classic example is head motion and schizophrenia. They're highly correlated. So if you very strictly exclude people with a lot of head motion, your schizophrenic sample will just really shrink. And so again, it's a trade-off. And the bottom line is, I think without interpretation, there is no science. So otherwise, we can just leave it to AI and it'll do a better job than we will. So we want to do something like statistical inference. And if we can't get all the way there, other ways like variable importance. So we want to do it at the right scale, for the right structures. And the most important thing is talk to scientists. Many people have already said that, but you have to keep talking to them and making sure that you do something that is relevant.
Thanks, Liza. Again, we'll have time for questions at the end. So our next speaker is Wes Thompson at the Laureate Institute. If you do anything with ABCD, Wes knows the answer. If you have any questions, just ask him.
I know the people to ask.
Yes, if nothing else, he knows who to ask. Okay, take it away.
Hey, thanks. Thanks, Tom. Thanks for inviting me to the session, and thanks to the organizers, Hongtu and Jian, and the other organizers. I'm going to talk about the kind of one thing that I I think I've learned from population neuroscience and something that I've tried to address the issue. I don't know, I'm sure there's better ways of doing it, but this is my idea. So I'll skip this one, everybody knows what F4 is, so just, I don't know if you all agree that there's been a replication crisis in the neuroscience literature, but there's at least been issues with replication. And it's been hypothesized that this crisis is some combination of small effect sizes, small sample sizes, high dimensionality, research use of freedom, and publication bias. I think, like, if you think about it, and if you're working with scientists where it's publish or perish, and you have these small data sets with a huge number of features, which have a lot of variability in the effect size estimates, that's almost like a perfect storm of combination of factors and moral hazards to find things that aren't necessarily there, or at least exaggerated. And it's been shown, at least in some areas, that brain behavior effect sizes are smaller in large population samples, like UK Biobank and ABCD. I think everybody's familiar with the Merrick et al. Paper. I think Tom and I are both co-authors on that. I don't know what you think, Tom, but I think they probably overstated the case to some extent. I don't think you always need thousands of people. You almost certainly don't always need thousands of people to do every analysis, but sometimes you do, and some effects are quite small. So this is just kind of a representation of what could be going on. So it's just on the left panel is just there's the true effect size is a absolute Pearson correlation of 0.1, and then there's a log 10 sample size on the x-axis, and then it just shows the variation in effect size estimates for those sample sizes. And the red mark is the 0.05 significance level. So everything above that is significant. So if you only publish things that have a p-value of 0.05 or less, you're going to have biased, upwardly biased effect sizes. The middle panel shows the bias as a function of sample size. So the red line is the bias. So by the time you get to something around 10,000, which is about the size of ABCD, the bias goes away essentially. And on the right side is basically a lit search where we looked at brain negative affect associations. You see a very similar pattern. It's not proof, but it's kind of demonstrates that perhaps that's partially what's going on. In addition to for individual effects being relatively small in a lot of cases, it appears that if you're working with large data sets, that the effects are not necessarily sparse, that they're widely distributed across the brain. So for example, one paper we published a few years ago using ABCD data found that ridge regression type methods outperform lasso in terms of out of sample prediction performance. So, and I've seen also with elastic net and trained on ABCD that essentially no sparsity is like, assuming sparsity does not improve prediction performance, it actually makes it a little bit worse. So in this scenario, this argues potentially for using methods that integrate effects across the brain, because if you integrate effects across the brain, you make it larger because you're integrating multiple effects, and also there's perhaps no true null associations in large sets of neuroimaging features. And this is precisely the scenario that the genome-wide association studies encountered over the last decade or two, where candidate gene studies failed to replicate. Effect sizes were much smaller than researchers had previously considered. And so there was this whole crisis in the genetics literature about where is this missing heritability? Because what we knew from twin studies that things like cognitive tricks were highly heritable, but they just weren't finding it in individual SNP or gene associations. So a very influential paper in 2011 by Yang et al. By Peter Vischer's group proposed something called GCTA, or genome-wide complex trait analysis. And this is just a way of assessing the total fraction of variance explained for all loci in the GWAS. And you can apply it to scenarios like in GWAS where the number of loci far exceeds the number of subjects. There's other FEE methods out there that are very commonly used. One is LD-squared regression that uses summary statistics, and then one that I've worked on with Armin Schwartzman at UCSD called GWAS, which nobody uses, but I just have to mention it. So, but anyway, so the basic idea is very simple. It's very closely related to ridge regression. So you have a vector Y of standardized continuous phenotypes. In GCTA, you could also have dichotomous phenotypes. X is a predictor matrix where N is the number of subjects and B is the number of features or loci. And these assume a linear model. But the one twist is they add like a prior and a beta that's normally distributed with mean zero on a variance convergence matrix sigma beta. In fact, it makes even simpler assumption that matrix is sigma squared beta times the identity matrix. If the variance if y has been standardized and so the variance of y is 1, then the fraction of variance explained by x for phenotype y is given just by h squared. That's called the SNP credibility, and that's just the number of features times sigma squared beta. And this was very influential because it was shown that if you apply this method to complex traits like height, like 40–50% of the variance of height is actually accounted for by the additive linear additive effects of common genetic variants. So it wasn't that they were looking at the wrong thing. It was just that the effects were tiny, widely distributed, and they were measuring them with a lot of noise, and they didn't surpass statistical significance individually. And you can also use this GCTA to make predictions. So you can estimate the BLUP. This is the usual formula for the BLUP. And if you have the BLUP estimated from a large sample, you can apply it to the genotype of somebody who a de novo subject from another, like you recruit afterwards, and you can get a genetic estimate of the genetic merit for whatever phenotype you're interested in. So you can use it for predictions. So I thought maybe we could do something similar in brain imaging. And so basically using GCTA type model. But it's a little unsatisfactory because the GCTA assumes that all the loci are exchangeable, so they all essentially explain the same amount of variance. And this ignores any kind of biological information we may have about the imaging features, relative importance of different features, or the biological characteristics of the features. And so I'm basically taking this model and making a simple adjustment. This is basically the same. This is the GCTA model, but I assume that for every feature, I have a vector Z of annotations. That could be anything. It could be continuous or discrete, or they could be genetic annotations. They could be whatever. But we have some biological information that we want to incorporate into the model that could relatively enrich or unenrich associations for features of that type. We don't know what that is, and so we have to learn it from the data. But we want to allow for the fact that like different aspects, different features of a imaging derived phenotype could be informative about how explanatory it is for a given complex trait. And so this essentially takes that beta prior, and they're still independent. H squared still has the interpretation of being the total variance explained by all features, but instead of the identity matrix, we now have this matrix P, which is still diagonal but has different weights depending on the annotations in this vector of alpha of weights that we have to learn from the data. So again, H squared is still the interpretation of the total variance explained by all the imaging features, but each feature is allowed to explain a different proportion of variance on average based on its vector of annotation Z and this annotation weights alpha, which we have to learn from the data. And you can get a BLUP somewhere as before. We place this into a Bayesian context by specifying priors on H squared and alpha, and put this into an MCMC algorithm, which is extremely slow, and so this is really not very practical at this point, and so we're trying to make it faster, but that's where it is at. Applying this to the ABCD study, everybody knows what the ABCD study is. So 11,880 children, ages 9 to 10 at baseline, and we just used the baseline data for this. We used the resting state MRI, and in particular, we used the Z transform Pearson correlations between the Gordon parcellations, which is roughly 62,000 features. Each of those, so that's each of those is an edge strength and correlation, and so we annotated each edge into a network, like so the 13 different networks from the Dosenbach et al. Paper. So for example, if the edge connected two regions which were in the default mode network, it would be default, so you could have any combination of those for the 13 different networks. And so those were the annotations for this model. We pre-visualized the data based for age, sex, genetic ancestry, and MR instance. I don't know if you can see this. The H squared estimate from this model was 0.37, meaning that the theoretically maximum amount of variation. Sorry, the phenotype was crystallized intelligence, but I didn't say this before. So the phenotype we're trying to predict is crystallized intelligence. So the maximum amount of predictive power we could possibly have if we estimated all those betas perfectly, if we had an infinite training sample, would be 37%. So that's kind of an upper bound on our ability to predict that. If you look at the annotations and the alphas, the alphas are on the first column. The only one that came out with a posterior credible interval that didn't cover 0 was default. And so this says that edges in the default mode network are enriched for associations above the general background edge strength in the data set. We computed the bluff from this, and then we again, using the training data was ABCD, and we tested the prediction performance in the human connectome project development, which was 220 subjects. The total sample size, by the way, is 5,643 after getting rid of people with high motion and getting rid of siblings. The test data had 220 subjects. The bluff using the usual GCTA with no annotations explained 11.6% of the variance in crystallized intelligence. And the bluff with the annotation-informed approach that we developed explains about 17% of the variance, which is in absolute terms not that high, but it's roughly 40–50% higher than in accuracy performance. Conclusions. Brain behavior associations have substantial similarities to GWAS in these large population data sets. High dimensional predictors, small but widely distributed individual effects. A simple exchangeable model like GCTA, it doesn't give you the capability of exploring kind of more biological in-depth biological associations. It just gives you one number, and so we're trying to add more biological ability to assess biological annotations, and incorporating those can potentially improve prediction performance out of sample. And I think I just have a couple minutes left, and so I just wanted to spend the last few minutes describing. You're all familiar with ABCD, but I want to describe another study very briefly called the Healthy Brain Child Development Study, which is currently running. The first data release for this is December of this year. The goal is to recruit 7,200 participants, 6,000 retained by the end of by the time the subjects are 4 years old. This is funded by the HEAL initiative, which is why it's capital HEAL. I know it's like stupid, but that's what they make us do. But it's like roughly a quarter of the participants. We're recruiting women during pregnancy and then following the children for at least 10 years. Roughly a quarter of the participants, the mothers have taken drugs during pregnancy. Another quarter are enriched for the risk of having taken drugs, and then roughly half the sample will be a population sample. Currently, we have almost 2,000 people enrolled. The first data release will have about 1,100 people in it. So the sampling is the protocol includes MRI, EEG, behavioral measures, and other measures. Let's see. So if we take a look at the kind of the visit structure, so visit 1 is prenatal. Visit 2 is the first postnatal visit, and that includes imaging. Visit 3, 4, 6, and 8 all include MRI. And then from visit 3 onwards, we'll have EEG, paired EEG as well. So I just want to advertise this is a data set that I think will be of interest to the field. I'm actually very excited about this data set. I think it's going to be, in some ways, a lot more interesting to analyze than ABCD because it's a very dynamic period of growth in the brain. And I'd just like to thank my funders and collaborators. Thank you.
US for staying time. We have time for one quick question. While we swap over. Well, let's just save it to the end. That's fine. Our next speaker is our esteemed organizer, Hongtu. We're to hear him talk about TBA. Oh, no. I'm sure that's a very important acronym to do with trans brain analyses.
I don't see anything.
Take it.
It seems like I'm the last speaker today. So that I don't have time limits, I can talk for 2 hours. Yours, my style is different from other speakers. Slightly. Yeah, what are the most important questions? And actually, I think the most important things these days people are doing. No, nothing wrong. Just because I didn't open. I think I quoted the phrase from the pro. The causality is not a mere useful concept. It's fundamental to our understanding of the world. Without a causal inference, we merely describe patterns, not explaining. And actually, I try to emphasize the most important thing these days. You think about the data science has been dramatically changing in the last decade. The most important things, we need to do a lot of deep applications, okay? Not just talk with the people, you need to do something deep than that. Another way is basically is statistical enough, of course it's not enough. Actually, you need a lot of knowledge from applied math, statistics, machine learning, and engineering. All are important things. And actually, when through my careers, when I first get into Columbia, basically psychiatry, and actually the most important things in my mind in the last 20 years is basically try to understand the connections, this path diagram from genes to brain disorders through this brain structure and functions. Okay, and that's have been in my mind for years. There's a lot of different things you try to connect them together. Okay, that's my key motivations in my career so far. Okay. The difficulty is you really have a data set to cover all these, important informations. But I think the whole world have been improving. So another thing is basically related to all these brain disorders. There's also multi-organs. That's basically we are these days. And actually, we're also interested in other organs in the body, and the brain, kidney, heart, retina, and all the liver, all the different organs. How could these organs interact with the brain in what sense? But that's something in my mind, have been for 20 years. And actually, to me, it's basically when we connect all kinds of informations, to me, there's a causal pathway from the genetics to imaging to clinical path. That's the most important things. And actually, these days, many psychiatric disease and the neurodegenerative disease using imaging as the biomarker, basically develop a treatment or intervention to treat the imaging biomarker. Because imaging is just a picture of the organs we care about in most cases across time. Okay. And actually, to be honest with you, these days, I read much more non-statistical papers than statistical papers, okay? And that's the reason how I find the second keynote, because it has a very nice review paper in Science last year. I told Jian, you need to get that person to give a talk. I think everybody benefited from his talk a lot. Right? And this is very important these days. I don't read a lot of statistical papers today. I just find something related to something I care about it. And also, I work with Jian on another big project on the study of AI. We try to assemble different data types, not the imaging. Imaging is one component of the big story in biomedical science. You see that we create this kind of data sets, and all the links to all these data sets. We also try to assemble the review papers on the topic. And another thing we are interested to try to link information across multiple different studies, like water biobanks. And in the genomics, that's basically they have the big dreams. They try to get the data across the world. Okay, that's very important, and for fairness and other important things. And Yuga Biobank is one of the pioneers for the study for that. And also people are thinking about how do I get the data set? To me, is I look around. I don't collect the data, right? And we have, friends basically collect ABCD data set. No, for me, I don't. And how to collect a data set? I go to the NIH data initiatives, try to get to the data set, and just understand the data set, and also get some other public data set. Another resource is basically you go to different dream challenges. They connect all kinds of dream challenge data set and they share with you. And also, these are great resources for us. And actually, when you have the data set, we have scientific problems, then you can develop methods, okay? And actually, there's a lot of methods can be developed because it's pretty open field and for statisticians. Because you think about statistics, maybe 10 years ago, before that, most of the method is a statistical method for structured data. But these days, we are looking for the method for unstructured data and also from the multiple sources. That's basically the current standards. And this is basically, I think that's the next 5 to 10 years, our whole society need to move forward on that. And how can the statistical community improve the processing and sharing the data? I quote Neil Bremer's phrase, we're in a period where there has never been such a wealth in new statistical problems and the source of the data. The danger is if we define the boundary of our field in terms of familiar tools and familiar problems, we'll fail to grasp the new opportunities. That's I learned a lot through my career by just try to understand this sentence, these two sentences, okay? And actually for neuroimaging data analysis in general, they have basically 4 major the tasks. One is deconvolution, structural learning, integration, and predictions, okay? And actually the image analysis essentially also statistics, and because you work on the data. In general, the image analysis have 2 major methods. One is deconvolution, another one is structural learning. Okay, the most of basically like image reconstruction. The typically is regarding using functional model, whatever simple models. That's the basically the parametric and non-parametric regression models. And also you can do imaging enhancement and image segmentation retractions. Okay, and I don't feel that imaging pre-processing is long statistical problems, okay? And if you're really working inside, you will see that this is typically statistical problem for each step, and actually just assemble them together. The image segmentation is the #1 problems in image analysis, okay, the most important things. And also another thing is just imaging registration, okay? And I just show you the tools. My friend Mark tried to develop a foundation model for segmentation. Actually, these days, segmentation is not that difficult. If you just use a unit and annotate the data, you can do by yourself, okay? The most challenging part is annotate the data, okay? And the technical is quite straightforward, okay? Be honest with you. And also you can do registrations and to speed up existing foundation models for registration. Also, the Mark also doing something, although my other collaborators working on this, how to develop foundation model for registrations, okay? And also there's a lot of related statistics, like use data imputation and basically using deep learning type of method, okay? And also other typically multitask methods for this kind of things, okay? And I think for imaging pipelines or like imaging processing, I think we need in our field or in general, and for the people working in the areas, we need a more good data pre-processing pipeline papers, okay? Really tell us what each step is doing. What's the fundamental logic behind it? That's so important. And also it's better basically we share the data processing pipelines, the good data processing pipelines with the societies. We try to basically build our own basic data pipelines and share with the communities through the state of AI. And also, you see that I read some of the Tom's efforts, the papers published in Nature. Okay, this is so important, the kinds of the effort to devote to do that. And actually, in the whole field, what are the problem we are facing these days is a good data set for comprehensive evaluation. Okay, for all these segmentation registration methods. Actually, there's no public available high quality imaging data set with detailed annotation information that cover a larger spectrum or segmented tasks in healthcare. Okay, and also a lot of statistical problems you can do it, such as to quantify the uncertainty and generalizability of brain disorder, brain analysis, as well as segmentation and regression tools. How to develop foundation model for various tasks. That's basically what people have been working. But I think this slide is basically the most important things for the whole community. And actually, how can we make a statistical method more impactful in the imaging communities? I tried to find a phrase. I can't, so that I asked ChatGPT to suggest the one. That's what the ChatGPT said. Okay, statistical method become truly impactful. We are not just about fitting our models, okay, and but about driving the discoveries, okay, by making the more complex imaging data interpretable, reproducible, and applicable in clinical practice. I think this sentence I really enjoyed it. That's the also is ChatGPT, okay. And actually, nobody has the time and also a lot of resources to do everything. Imaging is a great topic and it's broad enough. And actually, in my review papers, I cover some of these the major topics. And actually, yeah, if you're someone working in these areas, you can just work on one specific topic through your career. That maybe just should be enough. Okay. The most important thing, you do something really people like your method and use to solve real problem. That's the most important part. We don't need everyone to do everything. Okay, we just need someone to work on one of these topics, basically solved, really push the whole areas forward. Okay, this area is a great opportunity for everyone, in particular people from statistics and other data scientists. And actually, there are some other important topics I didn't cover, like interpretabilities, generalizabilities, and the data privacy, unbalanced data, and large language models. There's a lot of development along this direction, because imaging data is only one data type these days. And there's many other different data types. People try to integrate all the information together. And I just show you, basically, there's a great paper they published in Nature that quantify lifespan of the brain, okay? That's 2 years ago. The most important thing is even the model itself is simple when you look at it, but actually the problem is not that easy, okay? They spend tons of effort to sample the data set, process them, and basically harmonize them, and basically feed the model to that. Okay, there's a lot of statistical interesting problems behind it. And also, Tintin mentioned something about also lifespan, right? And the problem is not solved at all, because there are a lot of challenging problems behind it. And also, to me, in the field, if you really want to make a big impact, you need to know how you create some curated data set. Everybody else can use it. That's what would be extremely valuable for the societies. And also another thing is basically like Tom basically did a lot of contributing in the field because he developed some tools have been widely used by other groups. That's important. Another thing is these days we emphasize the products. Okay, it's not just about tools, data set. It's essentially you want to basically deliver kinds of the translations. Okay, and my personally, I really wanted to. There's a review paper called the Foundation Model for Generalist Medical Artificial Intelligence. It's a great paper, and basically, I personally wanted to build something around these lines, and also we're interested to integrate the data across different biobank data studies by using federal learning or other method, or through like the Enigma type of consortiums to do that, okay? And the last things, we set up this statistical AI analysis. This year, we have been working on a lot of joint efforts, and basically organize multiple events, and also share the videos about the education of statistics, and also the method development. And actually this year we give a panel discussion at the GSM is well received by our community. And also, because this is a lot of, because I'm the next editor of the JASA SAS, and actually we made a lot of new JASA initiatives. And there's two most important things. We have a new JASA SAS submissions. Basically, one is the short communications. And we have made a lot of change to our review systems in order to accommodate this. Basically, the principle is we want to cover the data infrastructures, data pre-processing pipelines, data annotation methods, comprehensive data analysis using familiar methods and perspective commentary papers. Okay, and I hope if you are interested, you can submit it, and then also you can discuss with us. Okay, even you have some paper rejected by top journals like Nature Method, Nature Genetics, we can chat. Okay, I can give a fast, basically, acceptance. Okay, another thing is basically we'll also welcome a limited number of review papers with a 50-page limit on major data type and applications. Okay, I list all these major topics I'm interested. Okay, if you are thinking you can find a group of top people to write, you're welcome to write. Okay, and thank you.
Excellent. Thanks all the speakers for keeping the time. So now we have time for questions. Anyone? I'll start one out for Wes. So Wes, is your approach with that phi, is that adding in addition to the genetics, or you're just saying using that approach to then detect associations with the number of variables instead of the genetics?
Instead of, though, it's you could use it in genetics also, but like I just applied it to brain imaging features, annotated brain.
Okay, so it's like looking for structure dependence induced by some covariates. That's a parsimonious way to represent this. Other questions? Eric?
Also a question to Wes. So you had those weights on the diagonal. Have you considered something off diagonal to take into account the kind of interrelationships?
Yeah, I haven't done it, but I think that's a good idea for certain features, because that's one of the differences between like brain imaging data and genetics. Like the GCTA assumes that all the effects are independent and kind of like sprinkled throughout the genome independently. And I don't think that's a safe assumption in brain imaging, because it's spatially correlated, and if you ignore that, you're at risk of inflating your heritability estimates. So something like that would be a good idea, I think. Yeah.
Questions? Yes.
So I have a question for Wes and also Hongtu on JASA. So it's a reproducibility. So you already mentioned, so the recent Nature paper say we need thousands of data in order to, so you say that we may not need that much. So what's a general, can you summarize what's a general approach to demonstrate?
Tom can probably say more than I, but I like if you're using like task MRI to predict the task that you're eliciting, you don't need thousands of people. You need like 50 people or something.
The specific that paper that drew all the attention, did try to be clear that it's about BWAS, so brain-wide association study. When you're trying to have an individual vertex or voxel find an association between a brain measure and some, frankly, noisy behavioral measure. So you get noisy brain, noisy behavior, or lifestyle, or whatever, and you need thousands of subjects to detect that. But in contrast, if you're trying to predict, for example, if you're trying to not do something brain-wide, where you're trying to get a map, but rather what combination of all the brain phenotypes will optimally predict something, it's very likely, if you've got good training data, you can have far less than thousands, or even just 100. So there have been a number of follow-up papers in response to that BWAS paper to point that out, that it's really about that brain-wide mapping question with noisy things. Because also, here's another brain-wide thing, mapping task activation. What parts of the brain respond to an in-fact working memory task? I'll take 12 subjects for that. That probably be enough, because that's the main effect as opposed to association.
But the key with the population neuroscience data sets is that they have a limited number of tasks that's intended to predict lots of different things that are not necessarily directly related to that task. And then that's kind of the BWAS situation. And that's what that America L paper was focusing on.
The question also for Hongtu on JASA reproducibility, any changes for that?
I think we added some new features behind it. Because for all these new type of submissions, and actually like the data processing pipeline, I think we have Zhengwu here, he's the AE for that, and he will develop protocol for doing it. Yeah, I think we will add more because the current reproducibility protocols that's not covered. We are going to do something. Okay, I haven't talked with him, but that's his job.
Hernando, would you want to unmute and ask your question to
Yes.
Lisa?
Thanks, Tom. Can you hear me?
Yes.
Yeah. Thanks to all the speakers for the excellent talks. My question is right now for Lisa. Lisa, thanks for the really great introduction to an overview of your work on networks. And in your talk, I think for the most part, you focused on correlation matrices, which are symmetric, positive, definite. Now, and then later on, you talked about directionality. And so if I want to consider a network which is not necessarily symmetric, so if I consider directionality as an important information in my connectivity matrix, what are the issues that I should be careful about when modeling these kinds of objects?
Right. I don't think I talked about directionality, but a lot of these methods, it doesn't actually matter whether the matrix is symmetric or not. If you're just thinking of edge weights as predictors and coefficients that you're estimating, a lot of the time having it asymmetric is actually simpler. You don't have to enforce symmetry. We work with mainly because it's resting state fMRI. We have to work with correlations, right? There's you can't align them otherwise. If it's task fMRI, you don't have to do that. Relating to the fact that it's a correlation matrix, there's all this work on using different metrics and treating them in their manifold space of semi-positive definite matrices. I've written a paper like that too. I found that in practice it matters a lot less than people who do that really wish for it to matter. And I think there's a very simple reason for that is because they are not spread all around this cone. They are actually kind of close together where Euclidean distances are not that different. So I don't really worry about the semi-positive definiteness at this point. I used to, but not anymore.
Okay, great. Thanks. Very helpful. Thanks, Lisa.
I have a question for Wes about the ABCD data set. So we use ABCD a lot. So one problem we found is that, we have a difficulty finding some data in some versions of the release. It seems like the latest release is not carrying some previous data. Is that true?
Which data are you having a hard time finding?
Like sleeping?
Oh, should be in there. Send me an email and I'll get it.
But I mean, I also want to hear some of your comments about, like you say you stop using NDA and try to move to another one. So can you explain more like what's the limitation of NDA?
Fernando can hear.
Oh.
Forgot to do it.
Yeah, so starting with the next data release, which is 6.0, it will no longer be through NDA. It'll be from a UCSD server through something called Deep. And so everybody will have to sign a new data use contract because it's a new system, a new validation system, and it'll be a new platform that's not NDA. Hopefully it's better than if it's not, we're in trouble.
So that cannot be worse.
Yeah, that was our philosophy. Yeah, but hopefully it's better. Again, if there's any issues, any problems, whatever, send me an email and I'll get it to the right person and I'll say it's not my fault. But yeah, I don't know if I can answer your question now, but seriously, send me an email and I'll be happy to answer it.
The price of admission. Personal contact with Wes and Thompson. Any last questions? I think we've basically used our time here. Okay, then let's thank all the speakers. And we can all retire downstairs where we have lunch. That's where the poster section is going to be. With